Welcome to Biosecurity Measures in the Poultry Industry, a qualitative review of biosecurity measures within the poultry industry. This is presented to you by Group B from the MPH 6006 class at Benedictine University in 2019. The aim of this study is to answer the research question, how do biosecurity measures in the poultry industry protect U.S. populations from salmonella outbreaks and epidemics? In this study, we are using a cross-sectional and ecological study design. An ecological study design comes from the ability for us to assess the exposure and risk factors over a given time period, as well as we now have a biosecurity measures focus using the U.S. human population as consumers of potential salmonella-contaminated products. From any gap in literature, we were able to find Salmonella enteritis is a public health hazard in the U.S. that can cause death to the immunocompromised. Prevention of Salmonella enteritis in laying hens is the first step in preventing an outbreak. By February of 2019, the CDC confirmed one of many outbreaks of multi-drug resistant Salmonella strains in the U.S. that were directly linked to consuming poultry products. Little is known as to why some birds can hold a drug-resistant strain of Salmonella, but one theory is a gene-resistant locus and a chromosomal locus. Our study will aim to analyze biosecurity measures and determine whether they are directly contributing to drug-resistant Salmonella strains, such as therapeutic or preventative antibiotics. Poultry, meat, and eggs are the most common source for human Salmonella infections. While Salmonella is not the most devastating disease among the poultry industry, it is the most difficult to control currently. Approximately 142,000 people are infected with Salmonella from chicken eggs annually, and about 30 people do die. The average American has 1 in 100 chance of contracting Salmonella from eating beef or pork, but they also have 1 in 40 chance of contracting it from eating poultry products alone. Salmonella can be carried in multiple ways. Some ways include chronic carriers, people who are immune to Salmonella bacteria themselves. These people only experience mild forms of the disease or even remain asymptomatic. However, the disease is carried over a long period of time. They are considered natural reservoirs for this disease. Although history has proven that outbreaks of Salmonella are the result of multiple forms of the bacteria, the most commonly seen are S. enteritis and S. typhimurium. This study aims to focus on certain biosecurity measures. Salmonella itself is often spread due to a lack of biosecurity measures. Anything like improving industrial practice, improving personal protection equipment, and other things around this involving chickens and Salmonella bacteria is really important to this study. A fun fact, roosters actually have a vertical transmission role in the transmission of Salmonella within chickens. Bacteriology samples collected by researchers concluded that Salmonella was among the top 5 isolated genera of bacteria among rooster semen samples that were collected. This concept would suggest that breeding hens would be inoculated with the bacteria at the time of semen transfer. Specified populations that are targeted within this study are families with children under the age of 5, individuals with weakened immune systems, farmers and industrial workers with chickens, meat packing employees, and federal agencies such as the USDA. Some reasons for inclusion for participants in our study might be that they might be employees of poultry processing plants, they may have received medical diagnosis of Salmonella infection, they may have presence of diarrhea or nausea and cramps extending over 3 days, they have an age eligibility over the 18 years age gap, they may have a primary caregiver for poultry species in question, and they may own chickens or ducks themselves. Some reasons for exclusion of study might be that they are under the years of 18, they may handle poultry only on occasion, they may have Salmonella exposure coming from non-poultry sources, or they may have obtained differential diagnosis from sources other than Salmonella. The study's targeted sampling approach uses aspects of stratified sampling, theoretical sampling, quota sampling, and even street ethnography. A systematic method in which controlled lists of specified populations within geographical districts are developed is one way that ethnographers map the targeted population. A known number of study participants are determined from the map made by ethnography. Quantitative data collected is done through questionnaires. Within these questionnaires, we will have ordinal and nominal metric measures. Some of these will answer specific questions, such as how often are proper hygiene practices followed by all staff within a chicken industry, perhaps. Other ones say, at any time during the processing of the animal, was there a point of cross-contamination? Yes or no? These kind of questions help develop qualitative and quantitative data and helps our data collection form into closed questions that are used so as to limit the answers provided. Quantitative data is also done via descriptive study. Descriptive study allows for in-depth research techniques, often following groups of people in or around infection sites, and can allow for further study over time. Other methods include epidemiological study, which can focus on mapping sources and sites of salmonella infections. With our quantitative data, we can then format correlational research. This allows us to investigate two variables that would be unethical to test experimentally. This also allows us to clearly see if there is a relationship between our two variables. What is the correlation between effective biosecurity measures and salmonella outbreaks? Unadjusted analysis will allow us to gain a better picture of the number of individuals that were infected by salmonella bacteria. Then we are able to format predictions. We make predictions about the effects of biosecurity measures on salmonella incidences. One example, such as local poultry processing plants, will be investigated in order to determine if lack of adherence to safety and biosecurity measures have contributed to outbreaks and recent incidences of salmonella within the community. This is how we use quantitative data to further our research. The descriptive analytics of the survey method are as follows. Demographic information will be collected, such as sex, race, education level, and other demographic features. General salmonella knowledge collected from survey questions provides us a lot of descriptive analytics. Generalized biosecurity questionnaire methods are also important for this study. Further, within the descriptive analysis, we have public health practices that are to be analyzed and then put into practice. To integrate certain qualitative measures, we can use certain techniques, such as associating certain breeds of chickens with specific outbreaks in the regions. Conducting one-on-one interviews with chicken owners and industrial places, we can use these interviews to establish a pattern of cross-contamination as well as geographical maps, and as well as to see where there is failure to adhere to proper biosecurity protocols. The most valuable asset to our study will be the specified case studies that look at specified groups of individuals to establish a robust model in an attempt to capture the common attributes of both animal and human data as it relates to the disease and its potential for outbreak. Finally, we'll be able to define our sample by finding exclusive members of certain poultry facilities. We can find farmers that handle poultry. We can find and refine our sample to only people who have been infected with salmonellosis. And we can have this group be our focus group, which allows us to efficiently get information and informatics from a number of people infected with salmonellosis. Final sample is best refined by the recruitment process using face-to-face interviews. This then uses qualitative analysis of salmonella detection, which will be collected from food workers, farmers, and people that handle poultry, and then people who have been infected with salmonellosis in any other way. This concludes our project on biosecurity measures within the poultry industry.